So we have another great lineup today, and we're starting it off with a session ambitiously titled How to Cure Rare Diseases. And so I'll hand it over to Dr. Karen Abraham of Tel Aviv University. She is a member of our Science and Medical Advisory Board. She was also an advisor for programming this conference. So thanks for all of your help in these ways and all the other ways that you help the GRI community, Dr. Abraham. So over to you.
Great, thank you. Well, here it's already late afternoon. It's really a pleasure to see all of you. I do wish, I have to confess, that we were together in Boston, but hopefully next year. Now, with your permission, before I introduce our speakers today, I'm actually going to give a shout out to Keith. And I'm doing that because if we were in person, this is something that we would do at the end of the conference, but I'm not going to get the mic at the end of the conference. This is my only opportunity. So you can blush a little, Keith, but I really, I know we all believe you are just such an inspiring driving force. Not only did you start CureGrin, but you are just an unbelievable cheerleader in the best sense of the word, and really keep us going. And you have incredible energy. So thank you. I know that I can hear everybody in the background, all the cheering and clapping that everyone is giving you. Okay, so as we've been working really hard, and we've heard a lot about it yesterday, and we'll continue to hear today about the research and all the work that we are all doing to try to figure out what the mechanisms that are involved in the GRIA disorders. What we really want to know is at the end, how can we find ways to treat and cure this very rare set of diseases? So we have some really wonderful guests today who have been doing this for quite some time. And so we're going to hear first from Dr. Michael Tranfaghlia, who is going to tell us about the work on Fragile X. And then we're going to hear from Dr. Bruce Blum, who's going to tell us about some other work that he's done in treatments for rare diseases. So let me start by introducing our first speaker. So Dr. Michael Tranfaghlia serves as the medical director and the chief scientific officer of FRAXA. And he works to coordinate the foundation's research strategy and works with universities and industry scientists to develop new therapeutic agents for Fragile X, for autism, and related developmental disorders. Now, you will hear more about this. At FRAXA's mission is to find effective treatments and ultimately a cure for Fragile X syndrome. Just a little bit about Dr. Tranfaghlia. He graduated from Harvard University with a BA in biology, and then he studied medicine at the University of North Carolina at Chapel Hill and did a residency in psychiatry. So Dr. Tranfaghlia, thank you very much for joining us today, and I'm sure we're going to learn a lot from you.
Thank you very much, Karen, and thank you, Keith, for inviting me to this conference. This is really great. I love working with other rare disease foundations, and I think we can learn a lot from each other, and hopefully you can learn a little bit from me. So I'm going to go ahead and share my screen and show you my PowerPoint. All righty. So can everybody see that okay? There we go.
Yep, looks great.
Awesome. Just gonna move you guys off to the side there. So the title I decided on, whoops, title I decided on for today's presentation is One Weird Trick to Cure Any Rare Disease. So how's that for getting to the point? A little bit of clickbait here. And of course, as is now standard, you're going to have to sit through all the rest of the slides to figure out what that one weird trick is, but don't you worry, we'll get to it at the end. I am an expert in Fragile X and how to develop treatments for Fragile X. And I say that because I am someone who has made every mistake you can possibly make in this field. And I think that qualifies me as an expert, at least according to good old Niels Bohr. So I'd like to convey some of those mistakes and the occasional successes that we've had along the way. I'll tell you a little bit about FRAXA Research Foundation. We're a nonprofit that my wife and I started in 1994. So we've been at this now for quite a while. It's been more than 28 years. We're still run entirely by Fragile X parents. My wife and I have been with it since the start, but we also have a few employees who are also Fragile X parents. And it really gives people a special motivation, I think. We're dedicated to funding biomedical research, and we funded over $32 million in direct research grants to date. Most importantly, we're founded on the principle that Fragile X is a treatable condition. And I think that was something that was very controversial at first, but it's now widely accepted. And it took a lot of work to change that viewpoint. We are in this for two of the best reasons possible, and their names are Andy and Laura. Our son Andy is now 32 years old, and he still lives with us. Our daughter Laura is a female with a full mutation of the Fragile X gene. And fortunately, she's very, very, very mildly affected. I would say completely unaffected, but then you really never know, do you? She's perfectly normal and wonderful, and she graduated from college, and she's married, and she has her own house and her own professional career, and she's doing great. Our son, unfortunately, is quite dependent on us and still quite disabled. So just the basics really quickly about Fragile X. You can actually see the Fragile site right on the long arm of the X chromosome right there. So it's an X-linked disorder with highly variable penetrance. It's the first of the trinucleotide repeat disorders that was recognized. And of course, Huntington's disease is the best known example now, but Fragile X was the first. Basically, having the trinucleotide repeat expansion just silences the gene. So the people with Fragile X have a normal coding sequence of this gene, leaving the possibility of gene reactivation open, but they don't produce any of the protein. And it's the level of the FMRP, the Fragile X protein, that correlates with the severity of the condition. And fortunately, we've got good animal models, and we've had it for a while. Fragile X is important because it's a major model for the study of autism. Just about all males with Fragile X have autism spectrum disorder as it's currently defined. And if we look at the childhood autism ratings scores of a cohort of Fragile X subjects at the University of North Carolina, actually, we see that about a third of the patients meet full criteria for autistic disorder. That's the formally defined full-blown autism. But just about everybody else has elevated scores that by today's standards would qualify them for autism spectrum disorder diagnosis. So what are we trying to do? The best way to describe it, I think, was what Dan Vershbow, one of our board members, said a long, long time ago. He said, this isn't basic science. This is an engineering project. We are engineering a solution for Fragile X, said the MIT graduate engineer. And Dan was a great guy who sadly passed away a few years ago, but we still follow this ethos. We are trying to engineer a solution for Fragile X. And I get the impression that the Cure Grin folks are like-minded individuals, and you're approaching this in a very similar way. So I think we have a lot of experiences that are relevant for you. We pursue this strategy by what we sort of tongue-in-cheek refer to as the FRAXA method, which is that we fund basic research. Basic research comes up with findings relating to the mechanisms of disease. We fund translational research that tries to identify drug targets that you can actually address and try to remedy the situation. We then conduct preclinical validation studies in animal models to see if there's promise in these strategies with specific compounds. And then we hopefully take these things into clinical trials. And when I say drugs and compounds, it could be biologicals, it could be ASOs, it could be gene therapy, it could be all sorts of things. Numerically, it's mostly small molecules, but that's just because of the state of the technology so far. Of course, we didn't invent this method. This isn't specific to FRAXA or to Fragile X. This is in fact just rational drug discovery. So we're a nonprofit trying to do rational drug discovery, or at least trying to incentivize rational drug discovery. So maybe we can call this community-based drug development. So we're not just trying to advocate for other people to develop new treatments. We're actually directly involved in it. We're trying to take an active role in this process. Now, this still requires pharma partners, because doing the whole job from start to finish as a nonprofit is just not realistic. I mean, just the regulatory costs alone of getting a single drug to the market. Not the actual clinical trials, but just the regulatory costs are millions and millions of dollars. And so only a few of the really large nonprofits could afford to do this. Something like the ALS Therapeutic Development Institute, they could probably afford it, but it will burn your cash very quickly if you try to do it on your own. The secret is to partner up with industry whenever possible. And it's also important to achieve leverage from everything you do. If you give a grant to a researcher, it's in the hopes that they're going to get much bigger grants from other places like NIH or the Simons Foundation or other larger nonprofits. So our first big break happened in the year 2000 when PI Mark Baer and his really sharp postdoc, Kim Huber, made a discovery in the Fragile X knockout mouse. They had this process they were studying that was a form of synaptic plasticity called long-term depression that they knew was mediated by metabotropic glutamate receptors. And they knew that it was also protein synthesis dependent. And at the time, a defect in activity-dependent protein synthesis in the postsynaptic compartment had recently been described. And so Mark and Kim thought that their mGluR LTD might be abolished in the Fragile X knockout mouse. And lo and behold, they found that it was actually twice as much in the knockout mouse as they would see in the wild type littermates. So this is really interesting. And it's especially interesting because it's known to be mediated primarily by metabotropic glutamate receptor type 5, mGluR5. And every major pharmaceutical company in the world was in the process of developing mGluR5 negative allosteric modulators, or I often just refer to them as mGluR5 antagonists. So this is a drug class in development that was very promising for a number of indications. And all we had to do was latch on to this and ride it to a cure, or so we thought. So we tested these mGluR5 antagonists, and there's a drug called MPEP that was a prototype of the class. It's still available from scientific supply houses. There's a number of these available. But we saw on the left-hand chart, you can see a nice dose response curve that in Fragile X knockout mice, we could rescue their audiogenic seizures, which was a major phenotype at the time, one of the most reliable phenotypes then. And we could also rescue the hyperactivity in the Fragile X mice in a nice dose-dependent fashion. So we thought, this is great. We validated this. We started collaborating with a number of pharmaceutical companies. It wasn't as simple as just testing it in a couple of mice. We actually did millions of dollars worth of de-risking studies. And we managed to attract a number of pharma partners, large and small. The smallest one was called Neuropharm, a startup in England that was developing a drug called Phenibam for Fragile X. It was their number 2 compound after fluoxetine for autism. Seaside Therapeutics was developing a more advanced drug that they licensed from Merck. And it was also their number 2 compound where they were developing baclofen for Fragile X and autism. But most importantly, Novartis and Roche had some of the most advanced mGluR5 antagonists available. And they got interested because they had to. When the smaller guys got interested, they had to get involved. And there's a lot of this follow the leader in the pharma business. All right, oops, oh, sorry. So long story short, Neuropharm went out of business after a successful phase 1 single ascending dose trial because their fluoxetine trial failed. The easy one actually didn't work for them. STX-107 at Seaside Therapeutics did well in phase 1 studies. They went out of business because their baclofen trial failed to meet its primary endpoints. although arbaclofen is still rattling around as a potential treatment for Fragile X and autism. Just get my, okay. Similarly, there were big clinical trials for the Roche and Novartis drugs, but they actually came up negative. So how is this possible? Is there a problem with the method? I mean, this is the way everybody discovers drugs, and everybody seems to have an equal amount of trouble with this. And in fact, I think we could see that there are issues at every step of the way, and probably the most, well, and we've got a lot of other candidates in the pipeline, so we certainly hope that it's not an intrinsic problem to the way we've been doing things. But most importantly, we've recognized that this is an iterative process of development, and it's probably unrealistic to think that you're gonna hit a home run at the first time up at the plate. I mean, you're gonna have to refine your methods. And how do you refine your methods? Well, you refine your methods by doing this. And we have this cycle that we think of as a development cycle where we unearth mechanisms of disease, but then we identify treatment targets and do the preclinical validation leading to the clinical trials. And if it's successful, we all sail off into the sunset, but probably it's not going to be successful. But all of these mistakes made along the way inform the next time around. And in reality, we expect that multiple cycles are going to be needed. And the pharma people are always talking about shots on goal that are necessary to develop a successful product. Well, above and beyond that, what are the mistakes that were made in the clinical trial, and what can we learn for it? Well, our son Andy was in the Novartis trial, and we actually saw fantastic results, but that may have a lot more to do with Andy and the specific combination of medications that he was on. But one thing we did observe was that there was a gradual development of tolerance. It was kind of slow over the course of 3 or 4 months. And we weren't really sure what was going on, but we looked back to see, is there any evidence that there might be tolerance to these mGlu-R5 drugs? And in fact, there was tolerance observed in the very first preclinical validation paper ever published. And whoops, I was a co-author on that. So we actually had evidence right from the beginning that there was tolerance to these drugs. And this is a kind of complicated graph, but it just shows that if you administer the drug, well, let's say you administer just water for 1 day or 5 days, you don't really decrease the audiogenic seizures by any appreciable amount. If you administer water for 4 days and then MPEP, you get a great response. But if you administer MPEP for 5 days in a row, you actually lose a significant amount of the response, and that's this group here. So this group versus that group shows you a drop-off in effectiveness after just 5 days in the mice. And interestingly, it is specific to the Fragile X mice, because in the wild-type mice, you don't see that. So the wild-type mice don't seize at the same rate as the Fragile X mice, but they get a great response to the drug, and they don't seem to show any tolerance. And it's not just MPEP. We studied a number of different mGlu-R5 antagonists, and they all had the same effect. But finally, we got a break because we have all these other targets that we're developing, and we have a drug that performed really well in the mouse model that is a phosphodiesterase inhibitor. And I won't go through all these specifically, but let's just say it worked really well in a mouse model on behavioral tests. We actually looked at dendritic spines, and it rescued a lot of the morphology that we see in the brain. And then most importantly, we did a clinical trial fairly recently with 30 subjects. So this is a phase 2 study. And it was very successful, and everything got better across the board. So we had a widespread improvement in Fragile X, and this is now going into major phase 2B, 3A clinical trials with this company, Tetra Therapeutics, which has since been acquired by Shionogi. And just to show you the extent of leverage, now that Shionogi is in the game and has declared their interest in Fragile X and will be investing something like $40 million in Fragile X clinical trials. All the other Japanese pharmaceutical companies are playing follow the leader, and they're knocking on our door now. And so we're working with a number of other companies to develop treatments through these companies, as well as the European companies that we've already been working with, and a whole slew of American usually smaller pharma partners. So we got a few things right along the way. The biggest thing was to focus on Fragile X. We didn't get distracted by some of the associated conditions that come from pre-mutations, or the neurodegenerative condition that happens in males with large pre-mutations called FAXTAS, or Fragile X tremor ataxia syndrome. We focused on Fragile X because that was the most important thing to us. And we feel like you can only do so much. If you try to do everything, it just dilutes your effectiveness. We set out right from the beginning to develop actual therapeutics. We believe that you have to swing for the seats. You know, you want to hit a home run. You're going to strike out more often if you're swinging for the seats. But we think it's really important to not settle for just doing awareness or support or trying to train young scientists for some nebulous point in the future. We think it's important that we actually go after a cure for Fragile X. And another thing that we got right is that we actually funded a number of resources rather than just the typical RFA, RFP model where you request proposals and you make grants to basic research, basic scientists. We actually funded resources so that all the scientists who are interested in studying Fragile X could have access to the mouse models, fly models, reagents, antibodies, the MPEP that I mentioned before. I would actually take little vials of MPEP around to scientific meetings we had and just distribute it free of charge. There's no better way to get somebody interested in something than to just give them some free stuff. And we had it synthesized at a very reasonable price, so it made it economical to do that. And each of the researchers that we were funding wouldn't have to pay Tokris prices to get their MPEP. They could get the same stuff that we were using for free. And one of the best ways that you can use to draw people into the field, new people, as well as to get the other folks who are already in the field talking to one another, is to invite them to special meetings that you're having, like now, only hopefully in the future, in person again. Some of the most successful meetings we ever had were at Banbury, where we would have 36 scientists in a really intimate environment where they really couldn't get away, they couldn't escape, and they had to actually pay attention. And that worked out really well, although we eventually outgrew that. I think the Banbury meetings at Cold Spring Harbor Labs would be a great thing for you guys to do if you're starting out on a smaller scale. We eventually moved to larger conference centers where we had our FRAXA investigators meetings. And we now have Gordon Research Conferences for Fragile X. In fact, in a couple of weeks, I'm heading off to Tuscany for the Gordon Research Conference. So it's on again after getting canceled 2 years ago. Another thing that we did right was what I like to call rational repurposing. So it's repurposing, but looking at available drugs. And you're simply applying a filter as you identify treatment targets to say, are these treatment targets that are addressable with available medications? And we've identified a number of available medications using this method, things like lithium, minocycline, baclofen, lovastatin, metformin, and a bunch of others. And minocycline in particular is currently being taken by thousands of people with Fragile X around the world, and including our son, and it's been a real success. It doesn't fix everything, but it helps. Contract research is another thing that we did right, and we established a kind of resource for drug validation. Basic scientists can't necessarily do everything, and a lot of people aren't familiar with in vivo work in animal models. They may be working in hippocampal slices and not doing behavioral testing on mice. So we actually maintain a facility to do that. But other kinds of contract research, like working with Healx, as Bruce will tell you more about, I think, in a little while, it's been incredibly valuable to generate new leads. You can work with a contractor to get something done very quickly, very simply. Looking at the challenges that you folks confront, you need to have more mouse models because you have multiple conditions, multiple mutations, and those things can be generated very quickly by contractors, by CROs, or an academic lab working on contract, and then you can use the resources to distribute those models. So we have done a few things wrong as well. We've been too narrowly focused for too long. We got sidetracked by the whole mGlu5 experience. We've learned that FMRP does more than just translation. It's not just postsynaptic, it's presynaptic too. You know, there's a tendency to find something and then you stop looking once you've found it. But that doesn't mean there isn't more than one thing going on. And in fact, in Fragile X, it's not just in the neurons, it's in the glia too. And we now know it's not just in the brain, it's metabolism too in the rest of the body. There's abnormal insulin signaling in Fragile X. And FMRP regulates all kinds of metabolic proteins, not just dendritic proteins. So you get mired in orthodoxy and you have to always watch out for that. We also found that drug repositioning is much harder than expected. So here I'm making a distinction between repurposing available drugs and repositioning drugs in development. And drug repurposing is incredibly powerful. And I often say that failure is a prerequisite for repositioning, because Novartis isn't going to let you get your hands on their drug they're developing for Alzheimer's disease just to study it for your rare indication, because that could jeopardize their big program. But drug repurposing of available drugs is incredibly powerful and useful. And it's really a great way to develop your clinical trials methodology, because you have to do clinical trials to get good at clinical trials. And we also expect that you're going to need combinations of drugs. And with the help of Healx, we actually generated a list of major repurposing predictions, but we found that of all of these, Solindac was one of the most successful in actual in vivo testing, but they'd also predicted fenformin, and we were already working on metformin, which is almost exactly the same thing. So we were very encouraged by this, and we're doing clinical trials of some of these combinations now. But just to give you an idea, you can repurpose simple things, and this probably has some bearing on the mutations you're looking at, because A2A receptors pair up with NMDA receptors, and they associate with one another, and that's a regulated process, the amount that they associate with one another. But we can even show that in the Fragile X mice, we can rescue some phenotypes with caffeine. So you see that the knockout phenotype, say nest building, can be brought up to wild type levels with some caffeine, but it doesn't rescue everything. You don't get any rescue in open field or grooming behaviors. But if we then combine that with another drug, forgive the spelling, our scientist is in Latin America and Spanish speaking. So these are like the Spanish versions of caffeine and quercetin. If we combine caffeine and quercetin, we rescue all 4 of these major phenotypes in a testing battery. So we get a nice combination effect from some drugs that are not even prescription drugs. So it's the power of combinations. And lastly, I'd just like to finish by saying that, you know, there are some major pitfalls for rare disease foundations, and I only observed these by working with a number of different foundations and attending their conferences. But there are, number one is the usual suspects, that as rare diseases have become more popular, a lot of scientists are making the rounds to different rare disease foundations and essentially doing the same research project over and over again. You wouldn't necessarily know that unless you actually collaborate with some of these other foundations. Another problem is that a lot of wealthy donors only want their funding to go to what they think of as the top people. But the problem is the top people are already well funded, and effectiveness happens at the margins by bringing new people into the field and funding people who wouldn't otherwise be working on things. And another pitfall is that rare diseases tend to have a relatively small group of scientists that are interested in the condition. And if you don't work to bring new blood into the field, you'll just end up funding the same people over and over again. And not that the same old people aren't good, but you're hoping that if they're really good, they'll secure larger funding. So what's the weird trick? You got to fail faster. You got to spin this cycle as fast as you can. And you got to spin multiple programs in parallel. This is the way you get to a treatment for your condition, or in your case, multiple conditions. So you definitely need to have parallel programs going, but you need to think about how we can do the first clinical trial as soon as possible. How can we improve our trials methods? How can we improve the number of animal models available? That's how you get to where you want to be. That's the one weird trick. And I hope that you can learn from some of our mistakes, and just wanted to let you know that we're available anytime to talk about some of this stuff. check out our website at fraxa.org.
Mike, that was incredible. And I think there's so many take-home messages there for all of us as we're working to try to figure out how to get to these cures and treatments. We have one, I'll give you one quick question that we have in the Q&A, and then we want to move to our next speaker. So the question is that there are rumors from the early Fragile X clinical trials that failed to show improvement, but that many of the patients actually reported improvements. These had not been identified as primary endpoints and therefore didn't count as part of the results of the trial. Was this only a rumor?
Oh, no, not at all. In fact, there were a number of people after the mGlu-R5 trials, for example, who ended up having some of the compounds synthesized and did a little gray market self-administration. So there were a number of people who had some really remarkable responses. Our son had a fantastic response to mavoglurant during the Novartis trial, but we clearly saw a waning of that effect over time. I think the reason he had the response in the first place was because the combination of medications that he was on, which was allowed, but I think we had a fortuitous synergistic effect from some of the medications, including minocycline.
So even with these trials, if they overall don't succeed, they are personalizing if they're individuals who are doing okay or doing well with them.
Well, there are some methods for adaptive studies, and perhaps Bruce will touch on some of this. There are ways of accessing this, but what we think happened was that there's a problem with the drug class as the mGlu-R5 negative allosteric modulators. There were, as I mentioned, a number of drug development programs for a number of different indications, and they've all failed. And what that tells me is that this is not a viable target. And you run into this in central nervous system research a lot. Now, that doesn't mean that it's not a viable target for your indication, for example. This is obviously very close to what you guys are doing. NMDA receptors, for example, there are available drugs, but they're difficult to target because if you hit them a little too hard, you can get psychosis if you go one way, seizures if you go the other. So the metabotropic glutamate receptors were seen as a way of sidestepping that problem. And there is still some development going on, but there has been a major setback in the field, and the trials that have been done have failed all over the place, including for Parkinson's disease, simple things like gastroesophageal reflux, addictions, anxiety disorders. All of these things were considered to be practically a no-brainer for mGlu-R5 NAMs, and they've all failed. And so that tells me that there's a problem with the target itself and with the drug class. We are actually doing research to try to understand the mechanisms of tolerance, but it's been very difficult and it's been very slow going. There's no easy answer. It's not a simple receptor-based phenomenon. But if we could ever get to the point where we got the effect that we saw in our son over the first couple of months on mavoglurant and could sustain that, that would really be something, because it really got to the core of the autism features, the cognitive rigidity that we see in most people with Fragile X. Well, that just melted away, and he was able to go to restaurants and do new activities and didn't have the problems with transitions that he always has. So it was really kind of magical for a little while, for a honeymoon period. It wasn't quite the flowers for Algernon scenario where everything was better and then it all went away. It wasn't quite that dramatic, but we did get a little glimpse of that. And we're hoping that someday we can come up with a combination, another drug that can offset the development of tolerance or resistance, as some people would call it. But yeah,
okay,
there was
Mike,
a problem with the primary endpoints, and that's always an issue in clinical trials methodology. But we've gotten better as a result of these failures.
Great. Thank you, Mike. Okay, we want to move on to our next talk, so we give Dr. Bruce Blum a chance to tell us about his work. So Dr. Blum is the Chief Collaboration Officer at Healx, which is based in the UK, and their mission is to use AI drug discovery to develop new treatments for rare diseases. Let me tell you a little bit about him. Early in his career, he was part of the senior management at Bausch & Lomb Oral Care Division. As Director of Professional Relations and Clinical Affairs, he supervised all the clinical research, supported product development and international training, and worked with the FDA. He's also been the Senior VP for the Healthcare Professional Liability Divisions and he's been responsible there for all functional units. He was formerly the CEO of Goldman Philanthropic Partnerships, and where the mission was to accelerate the search for cures. He also founded Cures Within Reach that carries on that mission through repurposing research. Along the way, he's also started several successful entrepreneurial ventures. So Dr. Bloom, please tell us what you've been doing. It seems like a whole lot.
Thanks, Karen, and thanks, Mike. Mike and I work really closely together on a lot of things. My nephew, Alex, who's 40, also has Fragile X. So all the things that Mike was talking to you about around the Fragile X community are things I've been involved in as well. So I'm going to take you on a whirlwind tour of 3 different kinds of things just really quickly, and then I'm available after this. If it's impossible to cover everything in 25 or 30 minutes, but I'm happy to be a resource for the GRI community after this. So what I want to talk about today is small molecules, artificial intelligence, and drug repurposing, and a little bit about the regulatory process. So let's start with small molecules. So, when we talk about this, we're talking about organic compounds that you can take orally or have injected into the body that reach intercellular targets. We talk about these, and what's wonderful about them is you can change the shape or change them just a little bit and get all sorts of different things that'll happen. 2 molecules that look almost exactly the same, with one having a methyl and one having a phenol, will be totally different in the body, even though their structure looks almost identical. And chemists know how to fix these things so that we can make them do things that we want them to do. So when we talk about small molecules, there's 3 different classes, and Mike sort of referred to this, but there are drugs, and the difference, a drug is something that's been approved through a regulatory process. Then there's a nutraceutical. Nutraceuticals are the same kind of biologically active substances, like caffeine that Mike was talking about. It's generally regarded as safe. It's in all sorts of things, and you can use it, but it does have significant biological activity in the body. The issue between drugs and nutraceuticals is one is regulatory approved, and then you get it in a form where it has some consistency and quality, and you're never sure about nutraceuticals. But the good news about nutraceuticals is you can use them, and there's all sorts of ways to repurpose a nutraceutical in the same way you could repurpose a drug without all of the regulatory hassle to do that. And I'll tell you a quick story about that in a second. And then shelved compounds are, future drugs, the same kind of biologically active substances that just haven't ever been proven safe, haven't been proven effective for anything, but they're proven safe for human use. And so we repurpose all 3 of these kinds of things, and they have different routes to the repurposing or repositioning. And some take shorter time, some take longer time, some have better economic commercialization potential, and some have less. So I just want to talk quickly about repurposing, which is what Healx does. And I'm going to talk about the different ways that repurposing happen, and then a little bit specifically about Healx itself. So some repurposing happens by accident. And there's lots of evidence of this out there. Many of us know the story of Viagra, which was originally tested as an angina, a heart drug, but it didn't reach the efficacy that the company wanted, Pfizer wanted. But in the reevaluation at the end of the trial, they did find out about this specific side effect, and it became an erectile dysfunction drug. And after that, repurposed further as a pulmonary hypertension drug. And it even has the ability, it looks like, to maybe treat Fragile X and some other neurodevelopmental or neurodegenerative diseases. There's a condition called a neonatal hemangioma, where these kids are born with these big red, dark red areas of the skin that have lots of blood vessels. And the drug propranolol, which is a heart drug, was used to treat these kids. Again, it just was a sort of a lucky discovery along the way. And then one of my favorite ones is this drug cyclodextrin, which is actually a sugar molecule that's used as an excipient, a way to hold other drugs together in a tablet, was used as a control molecule, like a sugar pill, in a test of Niemann-Pick type C. And it turned out that the test mice didn't get better, but all the control mice that were taking cyclodextrin did. And it turned out that this is a treatment for that disease. Most of what happens in repurposing is traditional biology, where you start with a gene, you find out what protein it makes, you look at what target there is, and then you start to figure out, well, what kinds of drugs are likely to do that. And when I was at Cures Within Reach, we funded one on a rare white blood cell disease called ALPS. And for less than $500,000 in 3 years, we helped the researcher build a mouse model. The mice got better. Then he did a 6-patient clinical trial. All 6 patients went into remission. And it's now the standard of care for that and 4 other similar types of diseases. And, nobody makes any money on this. There's a lot of money that gets saved, but a lot of patients who would have died in their late teens and early 20s are now going to live normal lives for a very small amount of money in a very short period of time. And there's another disease called familial dysautonomia, which is a one gene defect where a protein doesn't get made properly, and it affects the development of the autonomic nervous system and then the functioning. And a lab at Fordham University, and along with us and a couple of other patient groups, managed over a period of about a decade to find a handful of nutraceuticals that had the biological activity to either upregulate a gene or change the shape of a protein so that we can get these kids that were having about 5% normal protein in their bodies to get them up to 100% and overcome many of the conditions. And they specifically picked nutraceuticals because there were way less regulatory hurdles, and much of this work was done without any actual clinical trials. So the work was done in the animal models, they showed efficacy, and then working with the physicians out in the clinical practice and the patients, they slowly began to use these and see the outcomes, take the tests. So they did sort of real world evidence clinical trials, and they could do that because these were all nutraceuticals. Then there's drug screening that a lot of organizations do, where you create an assay in a cellular model, and it either lights up if you make the cells better or you make the cells worse. And then you take different concentrations of already available drugs and nutraceuticals or shelved compounds, and you apply them to these, and you use a little microscope and robotics to see whether you're changing what's going on in the cells. And when you find some hits, then you can do some more low throughput confirmation and other kinds of research. I have seen this done hundreds and hundreds of times. It makes great sense. It doesn't seem like it is a really successful way. And I think mostly that is because it's hard to develop the right kind of assay that gives you the right information. And I've been involved in several of these where we got some early hits that didn't pan out because it turned out that the assay really wasn't where we needed it to be. And it's hard to create an assay like that in a single cell that end up relating to something as complex as the whole human body. Sometimes we do repurposing through clinical observation. Patients have more than one disease. They take a drug for disease A and it ends up treating disease B. And so physicians then begin to start using it. Sometimes it's just physicians struggling to find a solution will, make an educated guess at what class of drugs might help in a different kind of condition. Patients sometimes self-treat, over social media and patient organizations. We know that people share information these days. And so there's a lot of repurposing that happens sort of at the grassroots level. And then the last kind that I'm going to talk about is in silico, which means using computer models, in silico screening, where we use massive amounts of data to create artificial intelligence and machine learning models that can discover new biology and and help us to repurpose drugs. So let me talk a little bit more in detail about that. So on this slide, you see the traditional drug discovery process where you use traditional biology to discover what's going on in a disease. You finally get to a target that leads you to some drugs where you might get a lead candidate. Then you get into the preclinical phase, and if it looks really good, you go into the clinical phases and on to regulatory approval. If you want to use an AI approach, you can skip some of the discovery and target validation by using all the data that's already been created by lots of other people and quickly come up with some predictions that can go into the preclinical validation. What you see is you shorten the timeline in many of these cases, and you increase the probability of success. And Healx, before I joined them, got started with Mike and FRAXA to do Fragile X, and hopefully later this month or early next month, the first patient will be in our clinical trial will be dosed with Sulindac, which is the drug Mike was talking about, which we hope in combination with several other drugs will lead to a therapy for Fragile X and then maybe be useful in other neurodevelopmental diseases like that. And we think the artificial intelligence approach can accelerate the speed, increase the scale, and improve the probability of success of all of these things. So let me just give you sort of an overview of what, at least at Healx, this artificial intelligence model looks like. And in the center, you see all those dots and lines. The dots represent data points, and the lines represent connections between one piece of data and another piece of data. And the size represents how many other pieces of data each piece is connected to. So the larger the dot, the more other pieces of data this piece of data is connected to. And so our goal is to collect as much highly curated data, meaning it seems like it is highly valid and replicable data, because if you put garbage data in there, you're going to get garbage answers out. So you want to have really high quality data. A lot of it comes from what we call structured biochemical data. So these are databases that thousands, if not millions of people are working on every day, like Chembl and Drug Bank, where through the replication process, you get really high quality data that everybody shares. It's widely publicly available. We add to that some in-house curated data that we get from private sources where we make sure it's highly curated and we put it in there, but it's not publicly available. And we're either given that or we pay to license it in. And then there's a tremendous amount of data that's available in the literature that hasn't been curated and isn't structured in computer language data, the medical literature and other texts. And we use a program called natural language processing, which connects words to each other and begins to create curated, highly quality data that we can put into this knowledge graph. So those three sets of different kinds of data go in there. And then we apply artificial intelligence algorithms. Those are questions that we ask the data. And the data has information on disease and on drugs. And the goal is to start with a question about a disease and go through all this data, hundreds of millions of data points with hundreds of billions, maybe even trillions of connections. And the computer makes all those connections and remembers them. And then in the end, we try and crawl all the way through this data where we get all the way to a disease where there's some connection between any two data points that all connect all the way to the end where we get from a disease to a drug. And then we get this printout of drugs that might be related to this disease in some way. And then we turn those over to our drug discovery experts, our human pharmacologists, who then sort through these evidence chains, we call them, and see how strong they are. Do they make sense? Did the computer really understand what it was connecting to? If it doesn't understand that, then we retrain it to be better the next time and run it all over again. And in the end, we get a handful of predictions where the drug seems to be reasonably safe to give to patients, where there's a a new kind of mechanism of action that we understand, and maybe a series of drugs that all might interact with the disease in a different way. So a combination might really solve this. And then we start moving those into the validation process. And so what you see here is, how quickly we can do that. So that takes a period, depending on the disease and the number of outcomes we get, that can be anywhere from, 6 to 12 weeks up to 6 months to go through that whole process with the computer running everything, the humans working on it, putting the information back in, running it again, and getting to that final list of drugs to move into the preclinical phase. And then the preclinical phase can take anywhere from, 6 to 36 months, depending on the models and how quick and slow things go. And then, if we're lucky, we can move on to a clinical phase, sometimes as early as 24 months after we start working on a disease. So I just want to talk a little bit about why, as Mike mentioned, repurposing is a good thing for a group like yours to be thinking about. So it's faster to patients, at least it can be. You can get lead compounds to clinical trials in under 2 years, or if you're using nutraceuticals, it can even be faster than that you can start getting them into patients and seeing what kind of impact. Physicians can use them without clinical trial validation in some situations where the drug is safe. After clinical trial validation, you can use them off-label. And off-label basically means they're not regulatory approved, but there's enough robust information in the published literature that would support a physician and a patient getting together and deciding whether they want to use this. And for many of these, you want to move them to regulatory approval, and you do this through the 505(b)(2) process, which, if I have some time, I'll talk quickly about. There is some increased safety. We already know how these drugs work, or the nutraceuticals work, in a lot of patients. We have to be careful that, sometimes they move from adults to kids, so you don't know things, or into a rare indication that might be different than the normal population, but at least we know something about these drugs, which is different than any new chemical. They can be cheaper to manufacture, buy, sell, test. They're often available in lots of countries. And so that's great. And then there's lots of data for research around the kinds of things where we need for both standard and in silico kinds of research. And then there are there's some hurdles to get there. The hurdles are that often for drug repurposing, even though you can go through this process, there's no economic support for this, no financial incentive. As Mike mentioned, metformin's being tested. Well, that drug costs about $3 a month at its most expensive. And so if you're a company like Healx and you're going to spend $50 million proving that it works in Fragile X, how are you ever going to make your money back if it only costs $3 and you can buy from 100 different generic manufacturers? So drug repurposing is great, but often you have to change the molecule to actually make it better for the patients and to provide some way of getting exclusivity in the market so that you can actually sell it. So then the last thing I want to talk about is a little bit about the stages of drug development and the regulatory process. Keith asked me to quickly talk about that. So the word IND stands for an investigational new drug. That's what you, and I'll talk to you about it in a second, what you do to get started in this process with the FDA or others. And then you get to an investigational new drug through your clinical trials, a new drug application that gets approved, and then post-marketing. And so the FDA doesn't test the drugs, but the sponsors do. And the sponsors can be pharmaceutical companies, academics, governments, sometimes patient groups, and they submit the study reports. That's how you get a drug approved. And it can be a commercial company. It can be an investigator at a academic institution. You can even have an individual clinical doctor who does a single patient IND. And any of these are ways that you can do this kind of work. Once you've done the IND and start doing all the studies, you once you have all the data, you apply to get a new drug application, and you have to have all these kinds of data. There's quite a bit that goes into it, obviously, and all of this is gathered in the preclinical and the clinical trial stages of a new drug application. By the way, I sent Keith a 100-slide presentation that the FDA put together for patient groups. That's really helpful, and obviously you couldn't go through 100 slides in even an hour, but I asked him to have it available to distribute to you. It's really very self-explanatory and a good tool for you to use and get some experts to help you out in the areas where you don't really, understand it or you need some support. But it's a great way of learning a little bit about this. And there's 3 kinds of NDA reviews, new drug application reviews. So for a very new molecule, something that's never been in humans, you go through the 505(b)(1). For a generic application where a drug has come off patent and you're a company and you want to now make the same drug that the original company, you go through 505(j). And for drug repurposing, you go through this 505(b)(2) pathway. And what's interesting about this is you get to rely on information that somebody else has produced. So when, for example, if you're repurposing metformin, you can get the data that the original companies and subsequent companies used and gave to the FDA on safety and toxicology and efficacy, and you can use those to support your application. So it shortens the time frame and reduces the cost in getting your drug approved for a new indication. Outside the US, there's the European Medical Agency that does the same kind of work as the FDA. Some differences, some similarities, basically the same kind of thing that the FDA does. You have to know what the differences are, but it's basically the same kind of thing. Now the UK is no longer in the EU, so it has its own MHRA that does this same kind of thing. And then in Japan, there's a similar organization that does its work. So I know that was like a quick whistle-stop tour of everything that and I'd love to be able to spend time and talk to you about more of this, but we don't have time today. So Karen, I think there might have been a question that I saw pop up, but if not, I'm happy to take any questions.
Yeah, there are no questions, but first of all, that was really wonderful. Very helpful, I think, as we're all thinking moving forward, how we're gonna be able to get all these things on the ground. It seems that you have a lot of experience, but also I was enlightened by, it seems that you're willing to help. So especially in the networking session, you'll be around to answer questions. I did want to ask, you talked about the structured biochemical data. Has AlphaFold helped move that faster?
AlphaFold's a really interesting set of data. It doesn't help us because we don't do structurally based artificial intelligence, but for companies like Cyclica and many other AIs out there that have this additional data, this is really potentially going to be a game changer and really helpful in the future. And just to sort of, Karen, to say what AlphaFold is, it basically can predict what every kind of, the shape of every protein that could ever be made, what it would look like. And so when you're thinking of creating a drug that will go into a receptor in a cell, knowing what shape it is will often tell you whether it will fit or not. And so having a database that could quickly sort through all these shapes might be really helpful as you're doing drug repurposing and you know what receptor you have in your particular disease. So, yeah, and not only that, I think the fact that they're able to do it will then figure out how can we do that same kind of thing for other things. Maybe we can start doing them for receptors or other kinds of molecules. So really interesting work.
Wonderful, thank you. Okay, so I think our session turns into pumpkins now. We have to turn it over to the next session. I really wanted to thank you, Dr. Tranfaghlia and Dr. Bloom. It was fantastic to hear about all the work you've done, and it seems that you have such a good perspective on the ways that we can try to move forward. So I'm going to hand over the baton to the next chair.